So let us look at what we term as protein structure. Proteins are diverse in nature. We all know that. They are diverse for several reasons. One is that proteins actually serve different roles. And because of the different roles that they serve, they need to have different chemical and physical properties. So we have some proteins that are very simple, and then we have those that are actually highly very complex. Okay, so that therefore means that the structures will always be different. So what structures do we have? Of course, we shall say that we have primary structure, secondary structure, tertiary structure, and quaternary structure. What we need to do is to be able to know the examples of the proteins that fall under each of these categories, and also be able to relate that to their functions. When we say that a protein is having a primary structure, can you give an example? And why is it that that primary structure is actually very important for that protein? That's very important. So let's start by looking at this now. So the scenario goes as follows. You have a biology practical session where students were given to students were given a diagram of a protein molecule. They are asked to identify the different levels of protein structure. The teacher, of course, will have to make explanations to the students such that they understand what they are looking at, but also be able to relate what they are looking at with the functions of the proteins. So here, the teacher basically explained that the function of the protein will depend on the extent of folding and its shape. She also demonstrates that heat and acids can actually denature the protein. Students observe the changes of how this denaturation happens using white egg, basically albumin. So what was done is that this white egg was heated and then vinegar was actually also added. One student during this practical actually asked if only the order of the amino acids, if this order of the amino acids changes, does the protein still retain the ability to carry out its function, or does the protein function stop? That's a question. I don't know how you could answer that, student. And then you have another student who also noticed that some proteins actually worked as a group of chains, not as one. Okay, is that observation correct? And why is it correct or wrong? So, of course, for you to be able to answer those questions, you should be very familiar with what we term as protein structure. That's one. And then two, you need to know the different levels of protein structure. And then definitely, you cannot miss talking about the different bonds in the protein molecule, because that directly relates to the protein structure, or the levels of protein structure. And then definitely you should know that the sequence of amino acids actually determine the protein that you're going to have. And if there is alteration, there is a change in the sequence of this amino acid, you're definitely going to have another protein, and that protein may not function as the original one. So that means that you need to know how amino acid sequence influence the function of the protein. And if actually there is replacement of an amino acid with another amino acid in a protein chain, you can have situations where a disease manifests. Can you give an example of such a disease? The other point, of course, that is being illustrated very well in this scenario is an issue to do with denaturation. Does the extent of denaturation of protein actually, is it the same? Okay, is that What influences this denaturation? What is this denaturation, by the way? We need to understand that. And then we also have a question that relates to proteins being made up of more than one polypeptide chain. If you have more than one polypeptide chain, what level of protein or structure is this? Can you give an example of a protein with such a structure and what is its role? So let's roll. We have 4 levels of protein structures. Please remember them: primary, secondary, tertiary, and quaternary. These are quite simple, right? So we start with primary. Primary is very simple, just a sequence of amino acids gives you a primary structure. So, of course, you know what makes up an amino acid. You have a few amino acids, just one following the other. That's a primary protein, or basically primary structure of a protein. An amino acid has 3 components, basically. You have the central carbon. This central carbon, if you're going to do an amino acid, you start with the central carbon. You put the carbon there. So this carbon is connected to 3 things: the amino group and the carboxyl group, and then the side chain, or the R group. That is the amino acid. So this is basically, we can illustrate that here. So you start with the central carbon. The central carbon is connected to 3 things: the carboxyl group, the amino group, and then the side chain, or the R's group. 3 things. So the sequence of these amino acids gives you the primary structure. Okay. So each amino acid is connected to the other through peptide bonds, and we have talked about this. Since it is just a chain, it means this chain of these amino acids is very flexible. It can fold, and therefore you can get different shapes of these primary proteins. Okay, so we move to the next level. And because of the extent to which you can have this folding, you can have different proteins that fall under this structure, and therefore they can perform different roles, and we shall look at that shortly. So the second level are the secondary. It is the secondary protein structure. Here, we are now talking about folding. Okay, so from secondary going forward, we are talking about folding, but this folding differs with the extent to which you have the different bonds and interactions. Okay, that's very important. So with the secondary, you're having local folded structures that form within a protein due to the interactions between the functional groups in the backbone. So we have beta folded proteins and also alpha, or what we can term as alpha helix, which is this, and then beta pleated sheets. These are all secondary proteins. You're having secondary protein structure. The bonds that we have in there, for primary, we only talk about peptide bonds. For here, we are talking about the peptide bonds. Definitely will have to be there, but in addition to the peptide bonds, we are also talking about hydrogen bonds. So that means that secondary proteins are much more stable compared to primary proteins. Okay, so like I said, we have two types of folding. The alpha helix is, you have a coiled configuration of the peptide backbone. It's more spiral, and then the beta sheeted one, you have segments of a peptide bending at 180 degrees and folding back on themselves. And this is what gives them that parallel appearance that you see here. So, the primary structure influences the extent of folding because they're highly flexible. So what proteins fall under that, and how can we relate that to the function of those proteins? One is that in primary structure, that kind of bonding that we have is the one that the secondary structure will actually depend on. So meaning that, of course, you start with the sequence of these amino acids before this higher folding actually happens. And therefore, we are saying that the formation of secondary structure is actually influenced by the sequence of the amino acids which we find in the primary structures. Okay, so certain amino acids favor secondary structures. So that means that the extent to which we can have folding, whether beta sheeted or the alpha helix, it will depend on the amino acids that we have. So if we have proline, basically, this will favor the beta sheeted structure, because the proline will actually disrupt the alpha helixes. Okay, and then like we said, we have hydrogen bonds in the secondary, and that means that they are actually more stronger. Secondary structures are more stable compared to primary because of the hydrogen bonds that we have in there. Okay, so that means that the secondary structures contribute to stability and shape in the protein. So now, as we move to the next level of protein structure, we need to note that in the protein, we talked about peptide bonds. In the secondary, we're talking about the hydrogen bonds in addition to the peptide bonds, because the peptide bonds are the ones that determine or that hold the amino acids together. Okay, and then now moving to the tertiary, we need to recognize the importance of hydrophobic interactions now. Okay, so the hydrophobic interactions play an important role in determining the structure of tertiary proteins. So in this case, we still retain the other types of bonds, the peptide bonds, the hydrogen bonds, and then we introduce now the interactions. So these interactions can be the hydrophobic interactions together with the van der Waals interactions. But also, you will still have disulfide bonds and ionic bonds. These will actually know what they are. Ionic bonds, positive, negative. That's an ionic bond. And that's what you see here. Disulfide, you have cysteine protein that has sulfur. So that interaction between sulfur of one cysteine residue with another gives us the disulfide bond. And then the hydrophobic is the chains that we can have interacting, the hydrophobic sections or components of a larger molecule like this. That's when you can have that. And then we have the van der Waals forces. These are still between the chains of this molecule. Okay, so we can see that in tertiary we are having more interactions, and then in quaternary, now we can move to quaternary. Now, the quaternary will still retain the interactions or the bondings that we have seen in the tertiary. What is unique is that you have more folding here, more folding. And That is one. Two is that you're having many or multiple polypeptide chains. Okay, and hemoglobin is an example of a tertiary protein. So you in a hemoglobin molecule, so hemoglobin is a protein and it is a tertiary protein. So in a hemoglobin molecule, you have 4 polypeptide chains. 4 polypeptide chains. So these 4 polypeptide chains, you have 2 of them are alpha, and then 2 of them are actually beta subunits.